others. There's a billion different enzymes out there that do a billion different functions. Some are going to be more efficient than others. And that structure, yes. So then if you were to get into other parts of this class, I'm going to write this down, let's see if you were getting into a mutation that were to affect that structure, that enzyme, it would still work a little bit, but maybe it wouldn't work quite as well. Yeah.
Is it, after getting concentrated, so is there any way that they restore, like they get back to the way they were, like?
Yeah,
Or
so going
they-
back to what I thought you said, so basically if you reduce, so here's your substrate concentration going up and up and up. Okay? So forget this part of us, just stay down here, okay? So substrate concentration is going up and up and up, it's doing its job, converting C to D, C to D, C to D. So you're using up A to B, so in theory the concentration of A to B is declining. So then you're moving in this direction along that axis, right? So then it's going to go more slowly. All right, so in a sense, yes, you're restoring it. So yeah, you can fully saturate something, and then as you use that substrate, it is going to go back down, and then it can speed back up. It's going to reach an equilibrium.
Oh.
All right, more? Ask questions. It helps me do better, so thank you. All right, inhibition. So, and I'm leading up to something with this. This is going to be an example, and then I'm going to kind of give you a bridge to what I'm leading up to, and I don't want to lose anybody. So again, ask questions if I lose you. So enzymes can be inhibited. I just want to introduce the idea of an enzyme being inhibited. Enzymes have an active site. Other molecules can compete for that active site, or they can just get in the way of that active site. All right? Okay, and one case of that is antibiotics called sulfa drugs. You've heard of sulfa drugs. It's a class of antibiotics. And the way sulfa drugs work is they basically compete with PABA, the substrate for the enzyme. So bacterial cells make their own folic acid. So you know we can't make our own folic acid. We have to supplement it through our own diets. Bacterial cells have the capability of making their own folic acid, enzymatically making their own folic acid. And they do that. As part of that process, PABA is one of the substrates in the steps to make folic acid. Folic acid is an essential nutrient that they can make themselves. But sulfa drugs, the way sulfa drugs work, the way sulfa drugs inhibit bacterial growth is by competing with PABA for the active site on the enzyme such that it can no longer make folic acid, and it makes bacterial cells sick. It can't grow, it can't metabolize. Right? So this is a case of competitive inhibition, and just an example of inhibition. That you can have something that's not the substrate target bind to the active site of an enzyme and mess it up. Prevent its activity and stop that metabolic pathway. All right? And this is not a terrible analogy because the synthesis of folic acid is going to be an anabolic pathway. All right? But so you can have, and this is the rocky transition, and I'll try to make it clear. So I told you that folic acid is an anabolic pathway. Folic acid for a bacterial cell is something that it's making, something that it's expending energy to make. Okay? You can also have covalent inhibition. So I gave you the example of putting a sulfa drug into this cell, putting a sulfa drug into your system, so that that cell can no longer divide, reproduce, metabolize, and do its stuff, and you're killing it off if you have an infection. All right? So you can stop the activity of that enzyme that way. But sometimes cells, for good reason, need to stop their own enzyme activity. All right? And they can covalently modify an enzyme. And that can lead to feedback. So let's stick with the production of folic acid. So if I'm a cell and I'm trying to make folic acid, that's an anabolic process. The anabolic process is use energy or release energy. Use energy. Good. So it's expensive to make folic acid. The cell is going to expend energy to make this folic acid. All right? So here's your substrate. In this case, maybe it is PABA. Okay? And the cell has to go through various enzymatic steps to get down to different end products. Maybe folic acid, maybe an amino acid tryptophan, maybe something else. All right? So it goes through these trying to get from substrate A through B to product E. But I have plenty of E available in the cell. I have plenty of folic acid, I have plenty of tryptophan, whatever it is I'm trying to make, I have plenty of it. So, that product E could conceivably go up and bind to an enzyme much earlier in the chain. Okay, covalently bind to it and shut it off. Okay, so just to see if you guys are following me, this is a little rocky. Why would that be good? You don't make too much product, and why do we want to make too much product? Hmm?
Why don't we?
Yes, why do we not want to bother to make too much product?
Because we don't need that.
We don't need that, we don't want to spend the energy. Good. Okay, good. So, yeah.
So the cells, they change the configuration of the substrates?
Interesting. Now, what I'm getting at here, they could, conceivably. No. They're not going to really change the configuration of the substrate, though. Sorry, synapses, give me a second. Fine. Really what I'm talking about here more is covalent modification. Okay, covalent modification is the case where here you have an enzyme up here. All right, this blue arrow enzyme. E may bind to that enzyme in such a way that it prevents the activity of that enzyme, so that I don't bother to make all these steps. I don't expend the energy to make all these steps. Yeah.
So that self-regulation for these stuff?
Yes. Excellent. He said, is that like self-regulation? And I said, yes. That's a great analogy. Great way to put it. So we call it feedback inhibition, self-regulation, if you like that term. I like it too. Okay. So basically what the cell is doing, this particularly relates to anabolic processes. Relates to catabolic ones as well, but not as elegantly. I like to use anabolism because it's easier to understand. If I'm expending the energy to build a molecule, folic acid, end product P, whatever you choose, if I have plenty of that product, it may go back and negatively inhibit this enzyme such that it doesn't continue to spend the energy making it. And when we get into the middle part of this class, with respect to the tryptophan and operon, we're going to actively see how that happens. I'm going to go through that and show you step by step in the second part of the course how that happens with respect to the generation of amino acids. All right. Okay, questions?
People have one thing. What causes the inhibitor to bind to the enzyme? Like, is it just concentration higher, or is it
Excellent
what
question. So
concentration
what causes this to bind? So our inhibitor in this case is sulfur. And the real nitty-gritty chemistry, I honestly don't know. But if it's a competitive inhibitor, it's a concentration NCBS. So you're going to have a very interesting, and I'm sure this gets into pharmacology that I don't understand. There's a sweet spot probably in concentration where you don't put so much of your medicine in such a toxic, but just enough medicine that it most efficiently competes with your substrate to dominate the binding of that active site. So it probably is a concentration issue. It's probably a lot of things.
Pretty much made like an animal.
Yeah. Shape and structure. Yeah. Yeah, so here you have the sulfanilamide right there, and the tablet right there. They're gonna both want to bind to that fairly closely. I think a lot of those work that way. Okay. Okay, so let's go ahead and get into catabolism itself. All right. So, much of what we're going to talk about is with respect to chemoheterotrophs. Because that's the easiest way to think about it. And then we're going to talk about the breakdown of glucose. We're going to go through glycolysis and a little bit of detail. And we're going to finish up with the Krebs cycle in slightly less detail. Chemoheterotrophs, the breakdown, getting energy, getting precursor metabolites from things like glucose, amino acids, etc. All right. So metabolism can really be considered in 3 different respiratory pathways. So catabolism, the goal of catabolism is to make ATP. All right. And it really goes through 3 pathways. Aerobic respiration, where you're dumping all those electrons on oxygen. Anaerobic respiration, where you're dumping them on something else, but it's obviously something with a very positive reductive potential. So it wants to take an electron. Maybe it's not as good as oxygen, but maybe it's almost as good, like nitrate. respiratory means. Respiratory means is the electrons are getting dumped on some sort of an exogenous electron acceptor. And what I mean by exogenous is I mean outside of the pathway of the metabolism. Oxygen, nitrate, sulfate. Alright? So here you have your organic energy and electron source, glucose. Okay? And eventually, those electrons go through an electron transport chain. And the building up to that we're going to get to. Alright? Okay? That generates a proton motive force. Some of these terms, if they're rough and you don't remember them very well from your principles of biology, we are going to get through all of it. It will all come together by the end of today in Thursday. Alright? That proton motive force does oxidative phosphorylation to generate ATP. And if it dumps those electrons from that electron transport chain, it's dumped on oxygen, it's aerobic respiration. If they get dumped on something else, carbon dioxide, nitrate, sulfate, that's anaerobic respiration. So aerobic or anaerobic really just means oxygen or non-oxygen. But there's always some sort of a terminal electron acceptor. Alright? Okay, quickly. Anyone know what I mean by oxidative phosphorylation? Alright, so the point of this is generating the energy, okay? Taking that energy, taking those electrons and moving them so that we can generate ATP. Alright, and this is scavenging the carbon atoms in those precursor metabolites. Alright, so that organic energy fuels electron transport chains, it generates ATP through oxidative phosphorylation. They also can get broken up into different precursor metabolites, generating ATP through SLP, means substrate level phosphorylation, which once ultimately oxidized to CO2, and then biosynthesis. The precursor metabolites get eventually from this little arrow is alluding to the anabolism that comes out of catabolism. Keep waving. So this is respiration. Contrast that with fermentation. And the main difference in fermentation is it's just a more closed system. It's not a great analogy. Well, what it is that your electron acceptors are endogenous to the metabolic pathway. Pyruvate can be an electron acceptor. Alright? So here again, you have organic energy, glucose, whatever, grape juice if you're trying to make wine. Right? You're going to do that in lab. Okay? The carbon atoms get pulled off of that. In the same way, it goes through the same similar breakdown procedures. And that's substrate level phosphorylation, which I'll give you I'll give you a little more detail on in just a second. And then you end up with the precursor metabolites for biosynthesis and fermentated products, ethanol, etc. Lactic acid for anaerobic metabolism in muscles. And the main difference is instead of generating an electron transport chain, you're dumping your electrons on endogenous things like pyruvate. And pyruvate is the end product of glycolysis. It's just another sugar. They don't dump their electrons on oxygen, which is really energetically favorable. They dump them on pyruvate, which is kind of a fun take it. Not as good, not as energetically valuable, but you can still dump your electrons there. You need to dump your electrons somewhere. Alright, so that's just respiration versus fermentation. So these are catabolic pathways. So enzyme catalyzed reactions. So the reactant serves as the substrate for the next reaction, providing materials for biosynthesis. Okay, so let's just look at a very coarse level of this. Alright? So what catabolism is doing is breaking down these high energy molecules, like proteins are getting broken down into amino acids, polysaccharides, glucose, etc., cellulose, starch, get broken down into monosaccharides, gets turned into glucose. This goes through glycolytic pathways with pyruvate as an endpoint. Okay. You can do the acids. And all of these things eventually fuel into TCA. So you may have heard Krebs cycle, citric acid cycle, TCA, tricarboxylic acid cycle, all of these things. Let me just make a caveat, is you are studying in your book to support this. Your book refers to glycolysis as the Embden-Meyerhoff pathway. Anyone else heard of that? All right, me neither. I've never heard of the Embden-Meyerhoff pathway. A lot of metabolism throughout graduate school. I've never heard of the Embden-Meyerhoff pathway until I read that book. Most of you probably, pretty much everyone here has heard of glycolysis, right? All right. Glycolysis is what you normally hear, but as you're reading your book, don't be thrown off. It's the Embden-Meyerhoff in the book. All right, nothing tool, maybe that's a new terminology. So the point being is you can generate ATP through these things, and you're also generating a lot of, as you break these molecules up, and I'm going to show you in detail in just a second, as you break these molecules up, you're also releasing a lot of electrons, because these bonds get broken. As you go from 6 carbon sugars down to 4 carbon sugars, bonds get broken, electrons get released, and they get captured on things like NAD to become NADH, and FADH2. Right? And that's what eventually dumps its electrons into the electron transport chain. All right, so let's go through glycolysis. All right. So glycolysis is just the breakdown of glucose, which is a 6 carbon sugar. And in the end, it breaks it down into 2 molecules of pyruvate, 3 carbon sugars. And in the process, you end up releasing a lot of reduced molecules like NADH, and you generate a net gain in ATPs. So the way I might ask this on an exam, we'll step through it here in just a second, but the way I might ask this on an exam is, don't expect you to draw the molecules. I would expect you to know it at this level, to know the names, to know that glucose captures a phosphate from ATP to become glucose 6-phosphate, etc. And you definitely need to know that you're taking that 6 carbon sugar and it's branching out to where you end up with 2 3 carbon sugars. All right, it's a mass balance. All right. So I don't expect you to know the enzymes, even though if I did teach them to you, it probably would help because they're nice intuitive names. I don't expect you to draw the molecules. If you do, like say you're a biochem person and you do already know this stuff, great. I do think it's a better way to learn it. Makes it all of a sudden make sense. For the level of this class and the level of what I expect you to know, you have to know that for exam. Okay, so let's just step through this. Let's step through it and kind of see what we're talking about. So if we take one molecule of glucose, and the first step is to expand an ATP. So sometimes you have to put energy in to get energy out. So in this case, the cell is going to expend 1 ATP to make that glucose 6-phosphate. All right? And then it gets isomerized from glucose to fructose. All right? So you have an enzyme activity here to go from glucose to fructose. So now you have fructose 6-phosphate. It expends 1 more ATP to make it fructose 1,6-bisphosphate. So now you still have a 6 carbon sugar that's been phosphorylated twice. So at this point, this is where it branches. So fructose 1,6-bisphosphate gets cleaved into glyceraldehyde 3-phosphate and DHAP. DHAP, don't sweat it, it's just a little transition state so that you can end up with 2 glyceraldehyde 3-phosphates. Alright, so the point being is you're taking this 6 carbon sugar with its 2-phosphate and breaking it up into 2 3 carbon sugars with 1 phosphate. It's really simple math. Alright? So now you have similar, these mirrored processes going along. Alright, so glyceraldehyde 3-phosphate gets phosphorylated again. But when when it gets phosphorylated, it takes up an inorganic phosphate, alright? In the process of taking up that inorganic phosphate, it loses one electron that reduces NAD to NADH. Okay, so now you have this 2-phosphorylated 3-carbon sugar. It releases one of those phosphates, takes in an inorganic phosphate, which isn't inexpensive, and then when it releases it to bind to ADP to form ATP. So you get an ATP out of this, and then 3-phosphoglycerine. So now in this case, you only have 1 phosphate. So you have this 3-carbon sugar with a phosphate. That gets a summarized in a second. The 2-phosphoglycerate, it just rearranges. Okay. And then it goes down to hydrolysis and then phosphoenol pyruvate. And then it finally cleaves off that last phosphate just to be pyruvate. So you get an ATP. So, that one glucose molecule to get down to real 2-pyruvate is split 2 ATPs, but it's gained 2 ATPs back. The net gain was 2 ATPs, right? And it's also gained 2 NADHs. 2 NADs have been reduced to NADH. Alright, so let's see the summary of that. We'll type it out. Alright, so you have glucose plus expending 2 ADPs plus 2 inorganic phosphates plus 2 NADs gives you 2 pyruvates. Pyruvate is a really important precursor molecule for a whole lot of stuff. You end up with a net yield of 2 ATPs. You get 2 NADHs and 2 hydrogen ions. And that's the sum of glycolysis. All right, questions on that before I move on? Okay, so as I said on your exam, I could easily ask you to step through and name these steps. And what would be important for me is I would like to see that you understand that if I dephosphorylate, just for example, if I dephosphorylate 1,3-bisphosphoglycerate to ATP, please know at least that this is not going to have a phosphate attached to it anymore. It's going to lose one phosphate. 3-phosphoglycerate. Okay. Okay, so let's quickly tie this into the TCA cycle and we'll stop. Krebs cycle, etc., however you've heard that. So remember, the glycolysis stops at pyruvate. And the TCA cycle doesn't stop at pyruvate. It doesn't start at pyruvate. It really starts at this acetyl-CoA. So what you're doing is you're taking that pyruvate comes together with an acetyl-CoA. And in the process, you're taking a 3-carbon sugar and a 2-carbon, combining it to make a 5-carbon citrate. All right? And in the process of doing that, in the process of combining those 2 molecules, you release acetyl-CoA, excuse me. You go from the 3, forgive me guys, just messed that up. 3-carbon pyruvate releases the CO2 to get to acetyl-CoA. Sorry. Okay? So it releases that CO2, or it releases that CO2, those hydrogen, those electrons that were associated with that bond get dumped on NAD, form NADH. Man, I've been doing this since I was 23 and I can still mess it up. Okay. So acetyl-CoA is a 2-carbon sugar. This is a cycle. This is a continually turning loop. So that 2-carbon comes in and binds with oxaloacetate. Oxaloacetate is a 4-carbon sugar. That's how you get the citrate 6 carbons. Sorry about that. That's where I messed up. Okay, so acetyl-CoA binds with oxaloacetate to form citrate. All right. So did I summarize the isocitrate? All right. Isocitric acid, and citrate loses a carbon in CO2, and in the process allows you to reduce NADH to NADH. The alpha ketone also helps form things like nucleic acids, ATP, nucleic acids, other bases. Okay, so you end up with 4-carbon succinate. Alright, up to fumarate. In the process of going to fumarate, you reduce FAD now to FADH2. And then the malate goes to oxaloacetate, and this malate converts to oxaloacetate. Still 4 carbons, but it releases another electron to reduce NAD to NADH. Alright, so let me tell you what I expect you to know in the test and the take home. One, obviously, since I budgeted in the beginning, I don't expect you guys to have this memorized on a test. I never expect you to spit this out on a test. I do, however, like much better that your book presents it to you with the molecules in hand, so you can see it and it's much more intuitive than just their names. But the important take home from this is that you're basically taking a molecule and altering its morphology and releasing carbon atoms, and every time you release a carbon atom, it gets oxidized down into CO2. You take the electron from that oxidation step and you funnel it onto something like NAD or FAD, etc. Okay? And so you're generating all these highly reduced compounds like NADH, FADH2, that are later on going to fuel the electron transport chain. Those electrons are going to get dumped onto the electron transport chain. It will give you on Thursday, and that will fuel further ATP synthesis. So it's important to understand this from the point of view of it's continually turning cycle. It's going to come up again on Thursday, even if I don't ask you to memorize this the way it is, it will come up again on Thursday. Because the difference is, and let me just say this, and I'll say it again on Thursday if it's not clear now, there's an important difference I'm going to make then, is the difference between oxidative phosphorylation and substrate level phosphorylation. So here you have a case of substrate level phosphorylation, but because these NADHs are reduced and are going to kill the electron transport chain, and eventually it's dumped on oxygen, that's oxidative phosphorylation. Fourth generation. We're all getting tired. I'll make sure you get it on Thursday. All right. At the end of this lecture, I put a nice animation from your book. It'll help. It is more detailed than I expect you to know for the exam.